Okay, I think in the interest of time, we will get moving. Good afternoon, everyone, and good morning for those that are on the West Coast. Thank you again for being here for another one of our virtual research showcase series, which is brought to you by the Dairy Farmers of Canada. This is a series that we'll be doing, have been doing for a number of months now, and will continue into the new year, and hopefully beyond that as well, to showcase for you the latest research, evidence, and where researchers are even going as we move forward in time here across a variety of different topics in the Canadian dairy industry. Today, we're really excited to bring you three excellent researchers and some of the notes and the research that they've been working on as part of the Cluster 3 funded project, which you see on your screen here, which is sponsored in part by the Dairy Farmers of Canada, their partners Lactanet, Novalet, the Canadian government through the Canadian Agricultural Partnership. I've posted in the chat, but for those of you that are interested in listening to the series of presentations, which are going to be presented in English, but prefer to hear them in French, you can just scroll down to the bottom of your screen to the interpretation button. Looks like a little globe. Click that and choose the French option. That will bring you to hear this discussion in or through our translator and our interpreter. Otherwise, you can sit back and listen to our three presentations. We will then have some opportunity for questions. My only other comment for you before we really jump in is to make sure that you are providing questions to us through the chat. That will get sent to myself and my teammate Carly. We will make sure to bring those forward for our researchers at the end of their talks to discuss a little bit more about your specific questions and comments as well. So with that, I think what we'll do is introduce our first speaker, and that is Dr. Simon Dufour. And a bit of an introduction for Simon. He is a professor of veterinary epidemiology at the Faculty of Veterinary Medicine at the University of Montreal. He currently leads the OPLE research group, and he's the co-director of the chair in biosecurity of dairy production. Simon is passionate about infectious disease epidemiology and about monitoring usage of antimicrobials in dairy animals. He also has a strong expertise in diagnostic strategies, control of infectious diseases, and development of data visualization tools for monitoring and benchmarking health issues. Today, Simon will be presenting his talk on the Mastitis Network, science-based solutions for the dairy industry. We're very excited to have you here, Simon. If you want to go ahead and turn your camera on, we will spotlight you here, and we can get going. Thanks, everyone, and we will talk to you at the end of our series of presentations.
Thank you, Stephen. I think my camera is already on, right? Can see it? Is it
good to go?
Yeah. Okay, good. Yeah, so well, thank you, everyone. So I'm going to present today about the Mastitis Network research program and some of the specific projects that we had actually under the Dairy Research Cluster 3. So the Mastitis Network has a research program that is covering 3 themes, so animal, pathogen, and the environment. So we're conducting research sometime on these specific themes, sometimes it's projects that are touching on many of these. And we have 9 different projects under Dairy Research Cluster 3. I'm going to be able to, well, together with Trevor and Jennifer after me, we're going to be able to present 3 of these projects actually. So I will present mainly on the other health relevance of different staphylococci bacteria. And then Trevor is going to present on how to best prepare cow for dry off when we're using automatic milking system. And then Jennifer on how we could possibly manipulate the microbiota, which is the population of bacteria and the udder, to prevent mastitis actually. And so these 3 research projects, they were conducted using a common research platform. So briefly, we had 5 herds using automatic milking system, roughly 700 cows that were followed. And because we're using these automatic milking system, we can have the quarter milk yield per day for these cows. And in parallel, we visited these farms every other week. For the whole lactation, every other week, we would take quarter milk sample from these cows, and also over the dry period, like you would see in the bottom. The idea is that we did some bacteriological analysis using Mastitis of these milk samples, so we were able to know what type of bacterial species were in there. And roughly a bit more than 25,000 milk samples were collected. It was between November 2018 up to March 2020. So as many of you had experienced it, we were shut down also by the COVID-19 pandemic. So I'll start with the other health relevance of different staphylococci, and then we're going to have Trevor and Jennifer that are going to follow. So regarding staphylococci, well, in the good old days, that would be pre-2015, when you were sending a milk sample for bacteriological analysis, you would receive results such as Staph aureus, other staph, Strep agalaxiae, Strep dysgalaxiae, Strep uterus, or other strep, and E. coli and Klebsiella. These were the main things actually that you were receiving as results. But nowadays, using Mastitis, it's quite different. So actually, this word cloud was made using the Mastitis results for about 1,100 milk samples that were collected in Canada, the US, and Brazil. And you see a lot of different words. And actually, Staph is really big in there. This is the most prevalent genus. And there are 24 different species, including Staph aureus, which is more known, but there are 23 others that we don't know much about. So the idea was, what's the relevance of these, well, let's say 23 Staph species, if we exclude Staph aureus that we already know about? Are they foes? Are they just irrelevant? It's just a finding, or maybe some of them could even be friendly for us or for the cows, could prevent other infection, or could, let's say, be associated with higher milk production. We don't know. So the idea was to try to look if the quarters that are infected by these species have an increase in somatic cell count. Do they have a reduction in milk yield? So basically, should we treat them? Should we care? Or should we even support these type of species if they are friendly actually for the cow? So the first question that we could answer was, what are the most frequent ones? So you see on this figure the results from our 27,000 milk samples. So almost 60% of the cows appear to be healthy. We have a bit of other pathogen. And then we have a number of different staphylococci. There's a long list, there's 24 of them. But you could see that the most prevalent are chromogenase, 5%, and then Epidermidis hemolyticus. And Staph aureus actually is not the most frequent, it's 1.4, but it's one of the common ones. So we could take this information and store it apart because it's a long list. Carly, you could possibly hit the, yeah, exactly. So these are the 7 most important. So we'll keep working. I'm always going to show all the results, but we're going to keep these 7 in mind as they are the most frequent actually. So the next thing that we look is, well, at calving in first lactation cow, this is the graph that you see on the left, and on the right in older cow at calving, what are the most frequent one that we're going to see? Is it going to be different? And you could see mostly a pattern that is similar, except, again, Carly, if you want to hit the button, Staph chromogenase, very, very frequent at first calving at the beginning of the lactation, so almost 10% of the quarter are infected, while it's not as frequent in older cows. And Staph epidermidis, this is another one that we just don't see in heifers, or not in these herds at least, but were frequent actually in older cows when they calve for the first time. So this is another piece of information now. We have the 7 most frequent one, but now we would also see that Staph chromogenase may be more of there's something strange about the first lactation cow, and Staph epidermidis seems to be more for older cows. So what about Staph chromogenase in particular? We had some study done in the past where we saw that how much of these we could retrieve from the teat skin. So not inside the mammary gland, but just the teat skin as function of their age. So starting from birth, 0, all the way up to heifers that is ready to give birth, 24 months. And you see on the y-axis the probability that the teat skin is colonized with Staph chromogenase. And you see actually that starting at around 12 months, you get 80% of the teeth actually that are colonized by this pathogen. So it seems to be something that is opportunistic. It can be part of the flora of the teat skin, but it was done in only one US herd, though it's not clear if it's the same everywhere. So what's the impact on somatic cell count? If we look at the first lactation to start with, you have the list here of the different staphylococci, the most frequent one. We've put it also the healthy quarter, the ones that were not infected, and they are listed from the worst to the best one. If you want to hit the button, Carly, yeah, exact. So now we could see that some of them, Simulans, aureus, chromogenase, seems to generate quite large somatic cell count. While Xylosis hemolyticus, it's fairly similar to a healthy quarter, actually. It doesn't seem to be doing much. And then you even have some like Staph hominis, where the somatic cell count appear to be lower in the quarter that are, we'll call it infected, but maybe it's not an infection, but are colonized with this bacteria. Now, if you look at second lactation, we have a pattern that is not exactly the same, but quite similar. Again, Carly, do you want to move on? Yeah, exact. So Simulans, epidermidis, aureus, chromogenase, Xylosis, all high somatic cell count in that case. Hominis, again, it doesn't seem to be very important. At least it's the same as the healthy quarter. And you have hemolyticus, maybe it's a wee bit higher than what we would see in first lactation, but it's not clearly a big concern for that one. So if we go back to what we know, Staph chromogenase, epidermidis, aureus, Simulans, Xylosis, all in red because they have high somatic cell count, hemolyticus, intermediate, and hominis clearly doesn't seem to be a big concern. The next thing that we look at is the dynamic of these infections during the dry period. So you have the proportion of quarter that were infected before drying off and the proportion that were infected after drying off. And you could see that hemolyticus, epidermidis, chromogenase, it seems to be reduced during the dry period. There's less of these infections, so it's a good thing. But for some of these other pathogens, actually, it's either increased or it's just maintained. The proportion is the same before drying off and after drying off. Now, what you're looking at right now is the proportion of quarter that are infected at both times. But are these the same quarter that stay infected, or did something else happen? And that's what we're going to see next. So actually, for many of these pathogens, during the dry period, it's quite a dynamic phenomenon. It's very unlike Staph aureus, where you have a quarter that is infected, it's still infected at calving. So for instance, on the left, you see that at dry off, we have a number of quarters that are infected with Staph chromogenase and a number that are not infected. Then as we move toward calving, we see that a lot of the infected quarter will be cured actually, but not all of them. But we will also have some new infection. So we had a, let's say, 6% prevalence at drying off, a 4% prevalence at next calving, but it's not exactly the same quarters actually that we were infected before and after. So, and this is a pattern that we saw for many of the different species actually. So in conclusion, we're starting to gain more knowledge on the relevance of these different Staph species. Possibly that many of them are opportunist bacteria. So they can colonize the teat skin, and if the conditions are right, they could possibly move to the mammary gland, and they will not always cause high somatic cell count. Some of them will do more often. Once in the mammary gland, though, there are some of these Staph that are as bad in terms of inflammation as what we would see for Staph aureus. So for instance, Xylosis, Simulans, epidermidis, and chromogenase. They are not as persistent, though, that what we see for Staph aureus in many cases. It's a work in progress. So actually, we still have work to do in the coming year. So we're going to be able to do the same work that I've presented here during the dry period, but for the lactation, actually, during the lactation of a dairy cow, and to look also at the persistency of these different infections during the lactation. And then we'll look at the impact on the quarter milk yield. Is there a reduction of quarter milk yield, and of the composition of the milk actually, was it changed? So this is some results that are, these are some results that we're going to have in the coming year. And well, there were many people involved. Maybe the most important one were Dahina, which was the PhD student doing this research, and Ariane, a master student that was also working on the same sampling. And some of the students also that Jennifer is going to present later. So that's it. I'll leave the floor to Trevor for next.
Wonderful. Thanks, Simon. That's great. And just as a reminder to everyone, as the presenters go through their work, by all means, please enter any questions that you might have for them in the chat. We will hold questions at the end and then have a chance to chat with all three researchers about those specific questions you might have. Up next, we have Dr. Trevor DeVries. Trevor is a professor and Canada Research Chair in Dairy Cattle Behavior and Welfare in the Department of Animal Bioscience at the University of Guelph. Dr. DeVries, his area of research includes understanding the effects of feeding management on the behavior of dairy cattle, the effects of early feeding management and housing on calf and replacement heifer feeding behavior, and the effects of housing and management on the behavioral patterns and risk of illness in dairy cows. He will be presenting his talk on impact of dry-off management and robotic milking systems on risk of intramammary infection today, and we will get his spotlight here for you. And Trevor, the floor is yours.
Great, thanks, Steve. Can you hear me?
Perfect.
Excellent. Well, yeah, thank you for organizing this, and thank you for DFC for sponsoring this program here today, and the research that we're going to be talking about. This specific project done as part of, as Simon described, the Mastitis Network's Dairy Research Cluster 3 project is focused specifically on dry-off management in robotic milking systems and relationship to risk of intramammary infection and udder health in general in dairy cows. This work, and I'll mention that off the top, is done in collaboration specifically with Dr. Simon Dufour, but also my colleague here at the University of Guelph, Dr. Dave Kelton, and also our colleague, Dr. Herman Barkma at the University of Calgary, which many people probably listening know all of those persons quite well. Some background to this project specifically, we know that dry-off management is really key for reducing the risk of intramammary infection during the dry period and subsequent udder health and production in the next lactation. And there's been much research on this over the years, focus specifically on how do we say reduce that risk, as well as reduce risk of things like udder engorgement, which we know is not only potentially negative from a health perspective, but also from a welfare perspective of those animals, whereby that can cause pain in those animals, particularly in that early post dry off kind of period. And so we know, again, that we can do that through management, say, including reducing milking frequency and energy or nutrient consumption of those cows prior to dry off. However, one of the challenges that we have is that for many herds, particularly our kind of small to medium-sized farms, milking with parlors, tie stalls, again, we can manage those cows individually a little easier. But specifically with parlor herds, that becomes a lot more difficult to implement. Unless we have, say, a large enough herd where we have the facilities that we've designed where we can have a dry-off group, and we're able to manage those cows maybe separately and do that on kind of a weekly basis. And so that got us kind of thinking from a research standpoint of where are there other opportunities in the industry to, say, do better when it comes to dry-off management. And robotic milking systems are definitely an opportunity. We know that the adoption rate continues to grow here in Canada at a dramatic rate, and we've seen that. There is more opportunity to, at a cow level, and even as Simone talked about, we can even look at things at a quarter level, specifically in robot scenarios. But from that cow's perspective, we have a better opportunity to, say, manipulate milking frequency, as well as even feeding management, recognizing that we're providing a certain proportion of nutrients, a small amount, but still a significant amount of nutrients to those cows through the robot, which is also acting as an attractant for those cows to the robot. And so if we're able to change permissions on, say, milking settings, feed settings at the robot, potentially we can do a better job in terms of bringing down, say, milk production prior to dry off and getting those cows down to a low level where they're not going to face udder engorgement following dry off, as well as reduce the risk of infection in that time period and over the dry period. We also know at the same time, and I should point out too, though, in theory, that's all very good. To this point, there had been no research on this. And so there's no real quantification of how well that can actually be done. At the same time, as I was just going to mention, we do know that the use of, say, dry cow antimicrobials and teat sealants can be more difficult in those systems, just based on kind of how we can access cows, and how easy it is to, say, even deal with things like teat sealants and such post calving in the next lactation. And so there's been some move towards trying to reduce our dependency on those for other reasons as well. I think that are obvious in terms of reducing our antimicrobial use from a prudency standpoint as well. And then the other thing that kind of came to mind when we were piecing this project together was thinking about what other factors maybe specific to udder health over the dry period in robot herds that we really haven't looked at, given that we don't have necessarily so much data related to robotic milking herds and udder health in dairy cows. So that kind of led to the objectives of the work that I'm going to share with you today. Specifically 2 objectives. One related to the project that Simone actually described already, with the 5 research herds in Quebec, or 5 commercial AMS farms in Quebec. And that was really to determine how reducing milking and feeding allowance in the weeks leading up to dry off in robot milk cows affects their milk yield and udder health, both before and after the dry period. And then we had a second objective, which was actually looked at with a separate data set, and that was to associate housing factors and management practices at dry off, during the dry period, and in the early post calving period, fresh period in those cows, on measures of udder health in that early lactation period. And so for our first study, and this is the study that was already described, we recruited and sampled about 445 cows from 5 robot herds between December 2018 and March 2020. And this study was led by the student that's pictured here. This is Ariane France, who was a master's student on this project and is currently working for Lactanet. Ariane led this part of the project, and what she did was assign cows, these 445 cows, to one of 4 treatment combinations. And that was basically having either their milking frequency restricted in the last 2 weeks prior to dry off or not, as well as having their feed allotment within the robotic milking system decreased from the current level that they were at, or not. And so we basically had a control group with no restrictions, a group with just milking restrictions, a group with just feeding restrictions, and then a 4th group with both. And so jumping right into the results, and this is just to show you, and I'll show you a few graphs that look the same here, but this just shows or verifies that our treatments worked, I guess, for example. And so this is actual milking frequency of the cows in the 2 weeks leading up to dry off. And so the acronyms on the bottom are non-decreased feeding, non-decreased milking, decreased feeding, decreased milking, and the combination of those 2. And what you see here is at the top line that's flat, that's our control cows that had no restrictions placed on them. So you can see that their milking frequency remained very, very steady across that time period. Those groups that had restricted milking frequency experienced a large decline across that 2-week period, as you can see on the bottom 2 lines. And then that middle line is those cows that actually did not have any milking restrictions, but actually had feed restrictions. And you see that even with the feed restriction in the robot, we see a decrease in their milking frequency. If we look at their actual feed allocation in the robot, this fits again with the treatments that we impose. So the top 2 lines are those 2 groups of cows that had no restrictions placed on them in terms of their feed allowance. Now, the amount of feed that they did consume did go down, potentially related to a decrease in visit frequency and decreased milk production over that time period that was naturally happening as well. But for those cows that did have feed restrictions placed on them in the robot, we see a larger decrease as signified by those bottom 2 lines. If we look at the actual, and this is what we're primarily interested in, is then how did we actually affect the milk yield of those cows leading up to dry off? What we see here is a nice decline in milk production across all groups. So even in the cows with no restrictions, we see a small decline in milk production, and that's the top line that you see there. Again, likely a natural decline in those cows as they approach higher days in milk and the end of their lactation. But for those cows that either had milking or feeding restriction, those are the middle two lines, we see a greater decline in milk yield. And for those cows that had both restricted milking and feeding settings in the robots, we see the greatest decline, with a drop of just over 5 kilograms of milk over that 2-week period leading up to dry off. Showing again, basically, or demonstrating that we could successfully reduce the milk production of those cows as kind of designed. Now, we didn't necessarily get down to some of the levels that we'd be targeting. Some of the previous research in this area would suggest that we need to get down cows to probably closer to 15 kilograms of milk per day to really have a beneficial impact from an udder health standpoint and from a reduction in udder engorgement standpoint in those cows. But nevertheless, we did impose or see that drop. Now, moving on to next slide, what we did observe across this time period, specifically at the time of dry off, as well as into the next lactation, was no negative effects of those treatments on the milk yield, milking behavior, or somatic cell count of those cows. And so I guess that's a positive from that perspective. Where we're still investigating, and Simon just presented in the previous presentation some of the results looking at, say, changes in bacteriology of the samples that we took pre and post dry period on those cows. We're still looking at that data and piecing that together to look at actual how effective those treatments were, specifically at a quarter level in terms of the development of new intramammary infections as well as cured intramammary infections in those cows, and whether or not we observe any difference between the treatments. And hopefully those results will be coming out in the coming new year or early in the next year. That gets us to the next study that we did, and this was done at basically at the same time. We were conducting actually a separate cluster study funded by the Dairy Farmers of Canada, looking at management on a large sample of robot farms from across Canada. And we were able to take a subsample of data from that study and specifically look at the milk quality and somatic cell count data of a large sample of cows from those farms. And so this was a study led by a current PhD student and former master's student of mine, Catalina Wagman, pictured here. And what Catalina did was take data from 166 robot farms from across Canada, and all those farms were enrolled in DHI milk recording at the time. We got all their milk recording data for that time period of October 2018 to September 2020, and had data acquired in terms of housing and management practices acquired through surveys that we conducted in both 2019 and 2020. From that data, and next slide, please, we were able to extract individual milk records for about 14,000 cows, on average about 84 cows per herd, ranging again from some smaller herds to larger sample size per herd. For all these cows, we had their DHI data for that time period, and we were able to isolate basically 14,000 dry periods in those cows. And so what we had is the somatic cell count data of those cows before dry off into their next lactation, their milk yield pre and post, their projected milk yield, their dry period length, and other cow level information, as well as herd level information on management practices on those farms. From that somatic cell count data, we looked at the somatic cell score of those cows, both prior to dry off and in the beginning part of their next lactation, and use that as one metric, and using their pre-dry off somatic cell score as a covariate to look at how that changed over the dry period. And then we also calculated, based on the somatic cell count as a proxy for intramammary infection, based on a cut point of 200,000 somatic cell counts, whether or not cows were not infected prior to dry off and infected or possibly infected post dry off based on their somatic cell count, and called that an incidence of a new intramammary infection, and basically summarized those data by cow and then at a herd level, to look at the average somatic cell count at a herd level, as well as the average incidence of new infections, both at a herd level. So jumping into the results, looking at the somatic cell count, what we did was then associate that data with various management practices, both prior to dry off and during the dry period. And this is just an example of some of the results that we demonstrated. So herds with higher average somatic cell count in the next lactation were associated with, for example, not separating their cows into a different pen in preparation for dry off. So not really having a planned a dry off kind of procedure for those cows, not using a teat sealant, not using blanket dry cow therapy. So again, those are things that we would typically associate with a risk of infection, risk of higher somatic cell count. So these things make sense. We also observed associations with management during the dry period. So for example, not leaving cows in the same group. So cows being moved between different dry cow groups. And then even in the early lactation period. So for example, putting cows on the robot immediately after calving versus maybe waiting a few days, milking them in an alternative format before going on the robot was also associated with higher somatic cell count. When we looked at this from a incidence of a new infection standpoint, some similar results and also different. Higher herd level average milk yield was associated with a lower incidence. So again, I think that again fits with the literature fairly well. Separating cows into a different pen prior to dry off, similar to the somatic cell count data, we saw that having some kind of preparation for dry off seemed to be beneficial. And then again, during the dry period, actually housing cows in pack pens and pens with stalls compared to only pack pens during the whole dry period seem to be protective for incidence of new infection. And again, we didn't necessarily have kind of cow level hygiene data and other data that we could maybe explain this result, but it could have to do with, yeah, just the overall condition of those facilities and maybe differences in management between those types of facilities on those farms. So just generally take homes from this work, what our first study demonstrated was that we could decrease milk yield effectively by reducing milking settings, feeding settings in the robot. So not surprising, but good to put numbers to. And like I said before, we're hoping to then also associate those treatments with infection risk and cure in those cows as well. And this is some of the first work to also just describe what are some of the risk factors associated with somatic cell counts and IMI incidents in these cows and robot herds, and specifically factors in and around the dry period. So grouping strategies, production levels, use of teat sealants, those kind of things, which again, shouldn't be surprises to us, but good to have numbers to support these things. So with that, again, a big thank you to all those that made this work possible, and particularly Dairy Farmers of Canada and supporting organizations to that. And I'd like to thank you for your attention and hope to have a good discussion following our next presentation.
Wonderful. Thanks, Trevor. That's great. Appreciate that overview and that insight as well. Up next, we have Jennifer Ronholm. And a brief introduction to Jennifer is here, I lost it for a sec there. Is Dr. Jennifer Ronholm is an assistant professor and William Dawson Scholar in the Faculty of Agricultural and Environmental Sciences at McGill University. Her research is focused on antagonistic interbacterial interactions, agricultural microbiology, and antimicrobial resistance. Jennifer is going to be presenting a very interesting talk on manipulating the microbiota to farm in the absence of antibiotics without consequences. So with that, Jennifer, I will hand over to you.
Awesome, thank you. I think I'm sharing my video.
You are, and here you are to everyone else.
Awesome, thank you. So next slide, please. Thanks. So when I used to give this presentation, I'd say that antibiotic resistance, the crisis was about to start or it was coming, but I think it's already reached international crisis status. In 2019, it was estimated that 4.95 million human deaths were associated with an AMR infection, which is a lot. And this is projected to raise to 10 million a year by 2050. That projection, the 2050 projection, was made in 2016, and there's every indication that post-COVID antibiotic treatments has sped this up. So we're going to get to the 10 million a year quicker. In terms of human health, these rates of infection and AMR infections are truly unsustainable. Given that 80% of antibiotics that are manufactured are used in agriculture, a really easy target for people who want to justifiably reduce AMR is agriculture. And a really naive solution to the AMR crisis is a straight up ban in antibiotic use in agriculture, although it is suggested quite frequently. But this is naive because it would affect animal health, and it would affect productivity and all, food sustainability. So if antibiotics were banned on dairy farms today, there'd be a huge increase in the rates of mastitis, and the resultant cost would be in the $43 to $73 a cow range. And this cost per cow would be very difficult for the average Canadian dairy farmer, possibly even fatal to the dairy farm. So next slide, please. Next slide. Thanks. So my lab works on removing the functional need for antibiotics by seeing if we can manipulate the microbiome and kind of make it another barrier to infection for pathogens. So to get there, what we do is we characterize the microbiome of healthy versus diseased cows, and we see if we can find bacteria that are more common in the healthy cows than the diseased cows. We attempt to obtain isolates of these bacteria, and then we try to use them to designer disease-resistant microbiomes that we can transfer to more animals in the herd to improve resistance to pathogens. So a big question is this feasible? Antibiotics are very good at removing infection, so is this actually going to be a good replacement? We know that the microbiome is a very non-specific barrier to infection. A good example of this is Clostridium difficile. No one really ever suffers from a Clostridium difficile infection out of the blue. It's generally because your microbiome has been depleted by antibiotics that you become infected. So even though we don't understand all the mechanisms here, we know that this non-specific barrier exists. But this barrier might actually be pretty specific. Several, if not all, pathogens we're finding out now encode virulence factors whose sole purpose is to modify or deplete the host microbiota and induce this state of dysbiosis. The state of dysbiosis helps the pathogen establish an infection and gain space and nutrients for its own infectious cycle. We also know that most bacteria in a host relationship or even in the soil or environment are constantly fighting each other for space and resources using all types of antimicrobial weapons, including organic acids, enzymes, bacteriocins, colicins, and type 6 secretion systems. So there are really specific examples of this, and a good one is Vibrio cholera, the causative agent of cholera, and Pseudomonas aeruginosa, which can be a pathogen in certain situations, but is also an environmental microbe that hangs out as a commensal in a lot of situations. These two can, coexist very harmoniously until Vibrio cholera is given signals that it's ready to have an infectious cycle. When Vibrio cholera starts its infectious cycle, it expresses a type 6 secretion system, which is kind of like a molecular needle, and injects the Pseudomonas aeruginosa with toxic effectors designed to kill the Pseudomonas aeruginosa. When Pseudomonas realizes it's under attack, it expresses its own type 6 secretion system, and in turn injects Vibrio cholera with toxic effectors that kill the cholera cells. What we don't know yet is if you have adequate numbers of Pseudomonas aeruginosa in your gastromicrobiome, are you resistant or are you even immune without your immune system to Vibrio cholera, something they're still working on. Another specific example is Listeria monocytogenes, which is a really common gastric pathogen in humans. When it gets into your body, it can go beyond your intestinal tract and cause abortions in pregnant females, or brain infections. One of the things that it does when it gets into the gastrointestinal tract is express a toxin called Listeria lysin S. Listeria lysin S doesn't kill all of the microbiome. It specifically kills Alobaculum and Alloprevotilla. And they were wondering why these two specific ones, but Alobaculum and Alloprevotilla are really good producers of butyrate and acetic acid. Butyrate and acetic acid downregulate virulence factor expression in Listeria and stop it from penetrating too deeply. So Listeria has evolved a system to get rid of the microbiome that might downregulate its own virulence. So the big question is in these interactions between pathogens and the microbiome, if the microbiome wins, does the infection fail? And very recently, earlier in 2022, we saw an example of where Citrobacter rhodium, which is a very serious gastrointestinal pathogen for mice, comes into the mouse intestinal tract and attacks E. coli to free up space and resources for itself. There are certain members of the E. coli species that also contain two different types of secretion systems. And when they're attacked by Citrobacter rhodium, they turn them on and kill very effectively the Citrobacter rhodium. And in this particular scenario, if these E. coli that have these two extra types of secretion systems are present, the mouse doesn't become infected with Citrobacter rhodium. So there are examples where very specific interactions protect the host from infection. So we wanted to look at this in cows, and we used the same samples that Simon collected. And of these cows, 342 incidences of clinical mastitis were recorded, 10 Staph aureus, 26 Klebsiella, and 25 E. coli. We're still working on the Klebs and the E. coli infections, but we do have good results for the interactions between Staph aureus and the microbiome. Next slide. Next slide. So 10 cows got Staph aureus infections. The red show a Staph aureus infection, the black show healthy milk samples, and the blue show mastitis infections that weren't caused by Staph aureus. We looked at the microbiome present in each of these using using 16S rRNA sequencing. Next slide.
I think there you go, Jennifer. You're just on mute.
Okay.
There you go.
Yeah, sorry, my internet became unstable. So we did a network analysis of the different organisms that were living in the healthy and the diseased quarters. And what we found was that certain bacteria did have very negative interactions with Staph aureus. So Aerococcus was the strongest negative interaction at the genus level. And then we also saw that Staphylococcus at the genus level had negative interactions with Staphylococcus. So to delineate further what was happening here, we did a little bit more advanced technique of shotgun metagenomics. Next slide. So looking at the shotgun metagenomics, we saw that you could have very high levels of Staph psilosis. And if you had very high levels of Staph psilosis, you were more likely to not develop a Staph aureus infection. And in some instances, you could be very, very colonized by Staph psilosis, 96%, but have a low somatic cell count, in this case, 59,000. We also saw that if you were colonized with Aerococcus ehrnachii, you were less likely to develop a Staph aureus infection. But we also saw that some Aerococcus ehrnachii seemed to develop high somatic cell count, clinical mastitis caused by Aerococcus ehrnachii. So how do we kind of like put together these facts and make this make sense? So next slide. So Staph psilosis is an environmental bacteria that we see in a lot of places. Simone identified it as something that was causing a lot of inflammation, but it's also commonly used in the food industry as a starter for milk and meat fermentations. But a couple studies have shown that it does actually cause inflammation comparable to Staph aureus in cow udders. Next slide. Aerococcus urinacii and Aerococcus verdens are extremely closely related. And the techniques we use to tell these apart, mastitis, probably mix up the two a bunch, and we should probably revisit the phylogeny of Aerococcus urinacii and Aerococcus verdens. But until we do that, I'm going to use Aerococcus urinacii to refer to both species here. But these are routinely cultured from raw milk from healthy cows, and they're routinely cultured from raw milk from mastitic cows. And in our study, we saw that they interacted negatively with Staph aureus. So next slide. So we have isolated Staph urinacii from healthy cows that seem to inhibit the growth of Staph aureus, even in a one-on-one system. So this plate shows Staph aureus as a lawn on which we've streaked our Aerococcus urinacii isolates, and they do kill the Staph aureus at this really reductionist ecosystem on a one-to-one. And some of our colleagues have seen the same thing with Staph psilosis, where it kills Staph aureus on a one-to-one ratio. But I think the question now is we have to figure out if something's happening at the subspecies level, where some Aerococcus urinacii and some Staph psilosis might be good for the udder and preventing infection, and perhaps other isolates at the subspecies level aren't. And that's our working hypothesis right now. So in the future, next slide. What we'd like to do is start pulling these isolates of Staph psilosis and Staph urinacii from healthy and sick cows, and seeing if there are genomic differences that would indicate at the subspecies level that some are good and some are bad. And put that in the mouse model as well to see if you do get differences in the sick and healthy cows be creating sick and healthy mice as well. What we also want to do is continue diving into the E. coli and the Klebsiella infections. And what we're seeing is Staph psilosis and Aerococcus urinacii are popping up there as well as possibly being preventive of Staph of E. coli and Klebsiella infections as well. So thank you so much for having me today, and thank you to the Dairy Farmers of Canada for funding this.
Wonderful. Thanks, Jennifer. I will get, if I can ask all of our speakers to turn their cameras on or keep their cameras on, just give me one moment and I will bring us all up here. Perfect. Okay, so everyone, you've had a chance to hear from our three researchers here. We've had a number of questions come in the chat, and about 10 minutes or so for us to get a bit of discussion going. Simon, I'll start with you. We had a couple of questions here. The first one was more practically focused. Through this process, did you come up with a practical way to collect milk samples in AMS herds?
Yeah. You go out there, you grab milk sample, you leave. Well, no, actually, there was a lot of concern before starting about how we would do that. And in our case, we had to catch a number of cows, not all of them, some specific cows in each herd, the one that were recruited as we were following them. And so we simply had our list of cow, and we were like three persons, sometimes more, moving together and just moving the cows into their into a stall, and actually collecting each of the quarter there. And I would say that 90% of the time it worked fine. And sometimes we needed a second person just to hold the cow so she doesn't back up while we're taking our sample. The cows in automatic milking system, I mean, Trevor, you're the behavior guy, but my feeling actually as a practitioner or a clinical person is that they are very, very calm when you're entering the barn. I don't know if it's the automatic milking system or it's just a coincidence, but they are less afraid of human. This is my impression, but
yeah, maybe I can add the one challenge here is that, yeah, we had to kind of rely on the time of the visit to make these samples. So something that could be looked at, again, this might be a whole other story, but if we have all the robot data from these cows, is actually to look at the time interval since the last milking or till the next milking, specifically related to that sampling, to see whether or not there's kind of any effect of that too, right? Because again, some of these cows would have been soon after a previous milking, some shortly before one, or maybe in between. So that could also have an impact here. and definitely as a potential limitation of this work, but again, something that we couldn't really get around, so to speak.
Yeah, there would be a number of cows that if we get there, a few minutes after she went out, then we would have to wait at the end of the visit to see if we could get some milk or just leave it for that sampling.
No, that makes sense. Another question focused on you, Simon. The average milk per cow at enrollment was around 25 kilograms per day. Was there a difference in milk reduction for cows milking more than 30 liters per day versus cows milking less than 25 liters per day at enrollment? In other words, did higher producing cows have a larger proportion or percent decrease in production versus lower producing cows?
That would be for Trevor, I think. That would be for the pre-dry work.
Yeah, actually, good question. In terms of kind of the proportional decrease, slightly higher, like the production, I guess, distribution around those cows was not maybe as wide as we would have maybe anticipated. So it was pretty consistent, and because of that, we were able to detect those differences between the groups. We did see a little bit, I would say, if we looked at kind of the distribution of those cows, particularly those that were on reduced milking and feeding settings, yeah, those cows with the highest level of milk at dry off, or sorry, prior to treatment imposition would have experienced the largest. Interestingly, when we looked at changes in somatic cell count on those cows, and this is where I'm hoping the bacteriological data on those quarter milk samples will shed some light on this. When we actually looked at maybe changes in somatic cell count and some preliminary analysis, it was kind of interesting is that the risk of infection seemed to be, at least from a somatic cell count basis, seemed to be greatest actually in our middle kind of production cows versus those cows on the very low end, kind of at the end, sorry, of the treatment period versus those at the higher end. And so that's something that we need to delve into a little bit more detail as well, hopefully with that quarter level IMI data.
Yeah, thanks, Trevor. I had another one pop up for you. What was the number of milkings per day used to reduce milking frequency for study 1?
Yeah, and so that was a challenge because we didn't necessarily, because we were doing this on commercial farms. If we were doing this on a research farm, we probably would have done this a little bit differently. But because we were on a commercial farm, we didn't necessarily want to impose too great of a decrease in milking frequency, such that cows might be at risk of not milking at all, based on fetching schedules that were done on those farms. And so the way we imposed that was actually basically the cows that had no restrictions, they could still milk up to presumably up to actually 4 times per day, as opposed to, or more, sorry, those that were on a reduced, they were restricted to 1, or if their expected milk yield per milking was over, I think 17 kilos, they could go twice. And that was to also minimize that risk of cows that were maybe higher producing only going once and actually facing kind of utter engorgement by not being milked. And that was a concern we had with the participating producers in that study.
Yep, no, that makes sense. Jennifer, a question for you. Is it possible that milk cultures with xylosis or Urinacea could be a result of these bacteria killing aureus enough to not find Staph aureus in the milk sample? This could create a false negative Staph aureus result?
That's an excellent question. I suppose that could happen, but I wouldn't expect it. If there's actually a somatic cell count that's indicating a Staph aureus infection, I would assume that there would be a Staph aureus culture present, regardless of what other things are there at the time.
Great. Simon, were the cure of staph infections during dry off spontaneous, or were they following dry cow therapy?
Probably most of them following dry cow therapy. We haven't dived into the detail, but we had 4 out of 5 of these herds that were practicing blanket dry cow therapy, so giving antimicrobial to every quarter of all cows when we did the study, and 1 herd used none actually. So we'll have to look to see if the one that survived were more specifically from the herd that did not use antimicrobial therapy, but that's a possibility. It's still a 75% cure rate overall, so it's quite large for chromogenes in that case, yeah.
Yeah, that's fair. Trevor, one more came up for you that I wonder if we just briefly touch on, sort of related to the previous question. What's known about the animal welfare effects of milk yield at dry off? And they provide an example here. So the treatments here reduced milk yield prior to dry off by about 5 kilos a day, yet the average yield at dry off was still around 20. Is this acceptable?
Yeah, and that's where I pointed that out when I was presenting that, is that some of the data that's out there would suggest that we need to probably get down to closer to 15 kilograms to really have potentially an impact both on the infectious status of those cows and the risk of infection, but also from an utter kind of pressure and engorgement standpoint. And so that's where a few of the previous studies have kind of pinpointed as where we need to get down to. Now, again, these things could be relative too, right? So even though we only got down to say 19, 20 kilos, is that better than that cow drying off at 35, 40 kilograms? And my assumption would be probably yes, right? So again, there's not necessarily even though some of that work would define kind of a lower number would be a nicer target, I would likely think that having some type of reduction, especially for those higher producing cows, is going to have some benefit from an engorgement and pain standpoint in those animals.
Great, thanks, Trevor. Jennifer, we had one more come up for you that I wonder if we could just try and squeeze in here. It's more from producers, just more sort of big picture thinking here. Is the work that you're doing or the subsequent work that you mentioned you're going to follow up on, do you see that as being critical information that can help guide policy or programs or even best management practices when it comes to this broader conversation around antimicrobial stewardship and really where the industry, how they need to sort of position themselves?
It's a good question. I don't really see this kind of work and the microbiome work guiding policy. I think it's more of a response to policy. I think the day is coming when we'll see more and more restrictions placed on antibiotic use. And I think the day will probably come where we won't be able to use many, if any, antibiotics in agriculture. And in preparation for that, I think we need to start thinking about alternate antibiotic agricultural targeted strategies. The things that we make for humans to deal with antibiotic resistance isn't necessarily going to work in cows. So as we start making more therapeutics to deal with this, I think microbiome manipulation is probably a really good targeted approach to agricultural infections, because you have such a, it's not like humans where everyone lives in a different house and eats different foods. You can actually attempt to manipulate the microbiome in these consistent populations. So
Yeah, that's great. Thanks, Jennifer. I want to thank all three of you again and everyone here for attending our session today. Just a final quick remark here. As we go into the holidays, of course, like most people, we will shut down for a litlittle bit. We will be back on January 18th to cover off another project under the cluster that's with Doctors Greg Keefe and Jay Trenton McClure, focused on extending cow longevity by improving calf management practices. Some really interesting research that we hope you'll be interested in attending a discussion on in the new year. Until then, thank you again, everyone, and happy holidays.